Yes, thank you, Mina. And I hope that I'll convince you all that that container media is its own animal. And so if we study production of crops in container media, it's going to have implication toward nursery ornamental crops. I'll talk about a little bit how the vegetable crops that we studied are kind of good case studies of lighter feeders and heavier feeders, for example. So hopefully there'll be nice implications for everyone. So I don't want to give too much of a recap of biochar. I know you've been learning about biochar basics for the last 2 sessions. In terms of container production and nursery crops, biochar has many potential benefits, including it can improve the air porosity, moisture holding, and nutrient retention. And so when we think about container media, we typically think of the 2 properties of air porosity and moisture holding and getting that proper balance between the 2. There's a bit of devil in the details here where for increasing air porosity, this really happens when you have larger particle size biochar, and it's going to have less of an effect if you have smaller particle size biochar. The moisture holding can be improved, which is funny that we're talking about improving both air porosity and moisture holding. So these larger, chunky, bigger particle sizes of biochar improve air porosity. Biochar also has numerous micropores, which are binding sites for water as well. So they can improve the physical air porosity, but they can also be sites for holding on to moisture as well. And then there's numerous, there's very large surface area with biochar particles. So, and these sites can attract cations or positively charged nutrients. So it increases the nutrient retention of our soilless substrates. Biochar itself can add nutrients such as N, P, K, calcium, magnesium, and some micronutrients. So just the nutritional effect of biochar can be helpful. Biochar is reported to promote microbial activity, and there's several modes of action here. One of them is supplying carbon, so there can be a food source for microbes. The micropores can actually be good sites for microbial development. And then if we look at microbial activity, it's usually kind of highest at neutral pH. And so some of our soilless mixes can have very low pH. And so if we increase the pH and we're somewhat closer to neutral, that can promote microbial activity. In terms of nursery production, biochar does have a greater bulk density, so it's heavier per unit volume than light substrate amendments like peat and perlite and vermiculite. So I always think there's some benefit, at least to a point, of increasing the weight of nursery substrates so that they hold plants more securely. Plants are less likely to topple over in wind, for example. As I'm sure you've learned all about in the last 2 sessions, all biochars are not equal, and they have various physical and chemical characteristics depending on the feedstock and the manufacturing conditions. And so those conditions depend on things like the heating time, the temperature, and the pressure that the feedstock undergoes. Here's just an example of many different feedstocks that can be used for biochar, diverse production temperatures that were used, and then pH and bioavailable nutrients that resulted. One of the things I was looking at is pH, which, as we know, is super important for maintaining a proper pH for nutrient availability. And pH varied from 5.1 to 11.1, just based on having these different biochar feedstocks and then production temperatures. The types of bioavailable nutrients also varied by biochar source as well. Here's an example biochar. This is one that was maple biochar. So in central New York, we have a lot of maple, which is a hardwood source. And this particular biochar was made at heating temperature of 1,000 to 1,200 degrees Fahrenheit. And then you can see the macronutrient content and the micronutrient content. So this particular biochar is very, very high in calcium, is the highest mineral element that it has. 55% of the mineral elements in total were calcium, 30% were potassium. So we've got about 7,500 parts per million calcium, 4,000 parts per million potassium. And then our other macronutrients, we still have a decent slug of magnesium and phosphorus, somewhat less sulfur. And then sodium, something we don't particularly want. It's not plant essential, and we don't want it to accumulate in the substrate. If we use the biochar as the whole substrate instead of as a substrate amendment, that sodium level might be a bit on the high side, but is a small fraction of the total substrate. Once you've amended it, it's not a huge issue. Then we also have some micronutrients as well. Aluminum would be something that we'd also want to keep our eye on, and we wouldn't want excessively high aluminum. So I'm going to go into a particular project that I was involved at Cornell. So this had us use greenhouse vegetable crops, as Mina mentioned. I'll mention first the kind of bigger picture that we were looking at. So our title of the project was Managing Organic Wastes through Pyrolysis to reduce fossil fuel consumption and increase sustainability in the greenhouse. So in this particular project, we worked with a local diversified farm, that they're Main Street Farms in Homer, New York. They grow vegetable transplants. They have their own high tunnel and field vegetable production and some hydroponic and aquaponic production year round. So they have one heated greenhouse in a double poly greenhouse. And in the case of this project, we were looking at heating the greenhouse with a wood furnace to reduce their reliance on fossil fuels or to save on their heating costs. We worked with a partner called ClearStack, which is a furnace company out of Connecticut that makes a low-cost pyrolysis furnace. And so this was their first installation into a commercial greenhouse with that pyrolysis furnace. So you probably talked a little bit about the biochar manufacturing process. Pyrolysis would be heating in the absence of oxygen or in low oxygen conditions, and you drive the combustible gases from the feedstock, and those get combusted to generate heat in this case, and then a lot of the wood residue is still left behind as charcoal or biochar. So this furnace was installed at the local commercial greenhouse. The initial objective was to look at wood waste streams that we knew were available in central New York. So they had access to municipal wood chips. We also had access to willow chips. So collaborating with a Cornell professor, Larry Smart, that has a willow breeding program. Willow is this fast growing tree that can be harvested every 3 to 5 years for wood chips. And so we tested their suitability. What we found, which maybe shouldn't be as much of a surprise in terms of using those wood feedstocks that we had available to us, is that it was all about quality control. And so in the end, we found that municipal wood chips were too variable in their size, and we needed commercial screening options to make them viable. So we weren't able to use this particular furnace with kind of the free municipal wood chips that we had access to. Moisture control was also really important, and so one would have to set up a large kind of system on your own operation to ideally cover wood chips avoid rain, so that they would have a low moisture content by the time they would get used in the winter time. The willow chips had kind of higher quality or higher uniformity of the chip size, and they were able to be used in the furnace to some degree. We didn't have access to them as a long-term um, fuel source, so we use them as an experimental fuel source, and you'll see me talking about their use as a biochar. And then we ended up, or longer term, the grower ended up using wood pellets. So a bit of a manufactured wood process that you have to pay money for, but it would be still cheaper than like fuel oil, for example. So that was kind of the big picture of this project. And then the idea is this furnace would heat the greenhouse, produce biochar, and then we conducted experiments on campus to look at the effects of different biochar on the resulting properties of like vegetable transplants. Right, and we wanted to quantify the heating savings that we could get. So this project, just to acknowledge the team that was involved, brought together a bunch of people, including soil scientists at Cornell, Janice Thies and Johannes Lehmann, an agricultural engineer, Michael Timmons, the Cornell Waste Institute, Jean Bonnetol and Mary Schwartz, and then economists. And then we had a postdoc, Jingjing Yin, who was our plant scientist that led the hands-on research efforts. Main Street Farms was the commercial grower, and ClearStack was the furnace provider, and Finger Lakes Biochar with Kathleen Draper helped in our setting up our project objectives and assessment. So that was the big picture. Then the piece that I'll talk about today, pertinent to our topic, is comparing different biochars as a substrate amendment in container production. So in this project, we grew lettuce, tomato, and basil. They're common vegetable transplants that, like I said, they can also be kind of nice indicator species. So tomatoes would be heavier feeders. They might show more benefits of increasing nutrients in the root zone. Lettuce and basil might be a little bit more salt sensitive, or sensitive to excessively high fertility. And then basil is also sensitive to higher pH. So if we had issues with higher pH, we would have expected to see those with basil. So the three types of biochar that we used one that we purchased that was commercially available, and some of the team members had previous experience with it as kind of considered a high quality biochar, and that was Black Owl. If you look online, I couldn't actually find what the feedstock was, so I guess that's proprietary, but some type of wood feedstock. Then we used mixed hardwood biochar. So our municipal wood waste has lots of mixed hardwoods, including maple in this area. And then we had willow chip biochar that we were able to access through that willow project. You can see they varied in their size. So we did grind and sieve them through a 2mm mesh. So we use the particles that fit through the 2mm mesh that were incorporated into the substrate. The substrate amendment test that we did, so we had a base soilless potting mix that was 75% peat and 25% perlite. And then we use different biochar application rates, so 0, 5, 10, and 20% by volume. So I was thinking about this for like the 20% by volume. So the mix had 20% biochar, then it would have had 60% peat and 20% perlite. We paid quite a bit of attention to pH. So the base potting mix with the base of peat that was unamended initially with limestone had a low pH of about 4. Then the biochars that we used had a pH of 7 to 8. So what we did is we prepared the different application rates. Then we measured the resulting pH, and then we added different rates of dolomitic limestone to get a pH of about 5.5. And I think this would be an important point for growers, is to be aware of the resulting pH effect of the different biochars that you use. We fertilized this crop in kind of a standard commercial fashion. So these were well fertilized with a conventional water-soluble fertilizer. That was a complete fertilizer, 21-5-20, and it was prepared at a common rate of 150 parts per million nitrogen. And in our common practices, the plants get the fertilizer on weekdays, and then on weekends they get clear water, which can help with flushing or reducing accumulation of soluble salts. We repeated each of the trials 3 times, so what we have are good statistically significant results. So we'll dive into the results. You'll see this for lettuce, basil, and tomatoes, and we'll have this series of graphs where we look at biochar incorporation rate on the x-axis from 0, 5, 10, up to 20% by volume. And then that was for the 3 different types of biochars that we studied: the commercially available biochar, the mixed hardwoods biochar, and then the willow chip biochar. For lettuce, a couple of the plant growth parameters were height and fresh weight. And if you're selling lettuce plants for consumption, not just as vegetable transplants, the fresh weight is what you will sell the plant on. So maybe the main points from these graphs is for willow chip biochar, there was a bit of an increase in height at 10% incorporation rate. And then for fresh weight of the plant, these patterns were not significant. We did see some benefit, at least in terms of that pattern, with 10% biochar incorporation rate for hardwood biochar and willow chip biochar, and actually a little bit of a pattern where fresh weight went down ever so slightly, but not statistically significant for the commercial biochar. Here's what the plants looked like with that willow chip biochar. So they were pretty large head stage when they were harvested. And we do see kind of this pattern with the willow chip biochar, where at, say, 10% incorporation rate, there's a pattern of plants being a bit bigger than the control plants that were at 0% incorporation rate. So to summarize the lettuce results, the willow chip biochar led to a bit of an increase in height. There was a pattern of a fresh weight increase from the hardwood and the willow chip biochar at 10%, and then that decreased a bit at 20%. So it didn't go down to below control, but it seemed like most of the benefit came at 10% incorporation. There were no significant effects from commercial biochar, but there was a pattern of decreased fresh weight at 5 to 20% incorporation. It was not particularly detrimental, but we normally would not want to see those decreases in fresh weight. Okay, then we'll look at tomatoes. For tomatoes, which we think of as a heavier feeding crop, if you look at plant height, notice that we see increased plant height at 5 to 20% for commercial biochar, and then especially at 10 and 20% for the hardwood biochar and the willow chip biochar. The plant fresh weight patterns were not statistically significant for hardwood and willow chip biochar. We did see an increase in plant weight with the commercial biochar. So tomato seemed like it was able to take advantage maybe of additional fertility that we had. Then this was interesting, and I don't know that I have a scientific explanation yet, but we did see when we evaluated the plants, we did see an increased flower number as we increased our biochar incorporation rate. So for example, for the commercial biochar on the left, we increased from 6 flowers per plant to 18 flowers per plant at 20% biochar incorporation rate. So something about the biochar addition, the fertility status of the plant, maybe there could be other microbial things going on, but that led to an increase in flower number. So to summarize the tomato results, we did see increased plant height with biochar at 5% for the commercial, or 10% for the hardwood and willow. Plant fresh weight increased with 20% of commercial biochar, and then some evidence for a fresh weight benefit for 10% hardwood and willow. And we did see an increase in flower number, which was quite interesting as well. Then for basil, and surprisingly, basil was our crop that actually showed the greatest benefits of biochar incorporation, at least from a fresh weight perspective. So there was a bit of a height increase for the commercial and the hardwood biochar. And then if we look at the lower set of graphs for the plant fresh weight, the commercial biochar gave us increased fresh weight at the 5% application rate. Beyond that, we saw that fresh weight started to go down. So it seemed like for basil, which is more sensitive to high pH and high salts, that more than 5% of the commercial biochar, it didn't bring down fresh weight below control, but it did show reduced benefits of biochar at those higher amounts. And then contrast that with the hardwood biochar and the Willocha biochar that we prepared in our study. And we saw the greatest benefits at a 10% application rate, where the fresh weight was increased by 50 to 60%, which is really quite dramatic. And so if we were selling our basil plants by fresh weight, we would get quite a bit higher economic return with the biochar incorporation. So we have photos of all the biochars for basil. So here's the commercial biochar, our control plant on the left, and the commercial biochar at 5, 10, and 20% by volume. Notice the increased height. You'll also notice increased branching with the biochar incorporation, and that was even more obvious for the other two biochars. So for the hardwood biochar, we had our greatest fresh weight at 10%, and you really see the increased branching that we get. So biochar might be increasing its nutrient status. I normally think of the fertilizer program that we used as being a very suitable fertilizer program and fairly optimum already. So I was kind of surprised that there was some additional benefit of biochar incorporation. And then with the Willocha biochar, we also saw the best growth at 10% and increased branching and greenness at those rates. So to summarize basil, we did see an increase in height with 20% commercial or 10% willow biochar. There was a fairly dramatic increase in fresh weight. It was a bit less dramatic with the commercial, that was at 5%. And then we saw the greatest benefits with 5 to 20% and really at 10% with the hardwood and willow. So the fresh weight gain was 60% for 10% biochar incorporation for the hardwood and willow, and there was no further benefit and some declines in that benefit at 20% biochar. Increased branching was apparent with the biochar addition, which I think would be an interesting phenomenon to further study. Overall lessons that we learned from our own greenhouse crop trials with biochar in soil as substrates were that responses to the plant varied by the biochar type and the plant species. So I think ultimately one is going to have to test biochars with your specific crop to make sure that there are benefits that are seen, or at least not detriments that are seen. We saw that a relatively small amount of biochar, often 5 to 10%, led to the greatest plant growth benefits, and often we didn't see additional benefits, at least in terms of plant fresh weight, with 20% biochar. And we didn't get to go in depth on the root zone nutrient status, but we would hypothesize that these growth benefits that we're seeing are either due to increased nutrients that we got from the biochar itself, or they could be due to the increased cation exchange capacity, and so the increased retention of nutrients that we had in the experiment. So in some of the growth benefits, there was a decline in those growth benefits at 20% biochar. And we hypothesized that could be due to increased salts, or like an increased EC of the root zone. EC being electrical conductivity, or a measure of the total fertilizer and non-fertilizer salts in the root zone. So our recommendations for nursery producers for using biochar in soilless substrates would be to, one, if you're getting a material from a supplier, is to request information and try to get as much information that you can about what is the biochar itself, what was the feedstock and its temperature, and then what are its properties. So we'd want to know its pH, its cation exchange capacity, its nutrient analysis, so the macronutrients and micronutrients. We'd also like to know its analysis for salts that we don't particularly want, like sodium and chloride and aluminum, to make sure that they're not going to be too high. And then it's helpful if a supplier has used it. It'd be useful to know if they've used it in soilless substrates in the past, and if they have recommended application rates. Then I always recommend that growers conduct their own small scale trials. So, we don't want to bet the farm on something that may have looked promising, or the supplier told us it was very promising, but conduct your own small scale trials first. So we want to make sure that the biochar is not harmful to the plants at the application rates that we used. And then test that it is beneficial. Are we seeing some particular benefit to justify adopting it as a substrate amendment? And then a big one, as I talked about, is the pH effect of biochars. Many of the biochars have a pH that is 7 or higher. In our soilless substrates, we typically want the pH to be between maybe 5.5 to 6, or 5.5 to 6.5, depending on the type of plant. And so if biochar is being amended and it's going to be increasing the pH of our substrate, take that into account, and that can reduce the need for liming. And then overall biochar adoption may require adjusting nutrition and irrigation practices. So if it's a material that holds more water than the other substrate components that you had, then you may actually need to irrigate a bit less. And so also account for the nutritional effects that you might see. So if we think we're adding lots of calcium and potassium, maybe we need to add a bit less of those in the fertilizer program for the plants. Okay, with that, I would like to stop, and it looks like a bunch of questions have come in through the Q&A.
Yeah, there are quite a few questions there. So Neil, if you want to open up the Q&A and just start tackling them.
Okay, that sounds great. And you could be my eyes and ears, Mina, if I forget something or need to expand.
Absolutely,
on
yeah.
So so one person asks, how can I produce biochar from granular sawdust at home? I'm not I'm not an expert in this area. This photo actually is a nice example of, you can get these biochar kilns that are basically a controlled way of burning a substrate, and the kiln helps you reach appropriate temperatures. There's different variations. They can be quite simple, they can be more complex. And then they would have instructions on, at some point, the way that we get the anoxic environment is by covering them with water. So there's a whole body of work on kind of simple ways that you can produce biochar at home. Does raising pH in the media to accommodate microbial activity not adversely impact nutrient uptake, is what Tom asks. I would say yes, that we have to make sure that we don't raise the pH too much. And that's one of the things that I've tried to point out as well. Like sure, we want microbial activity, but in the end, we're growing plants. And so, yeah, I think that's all I'll say about that. What are some good sources to look to when you are looking for lower pH biochar suppliers? I don't know that I have good information on that. And maybe we can see if some of the other speakers can weigh in over time on kind of feedstocks or processes that they've seen that result in a lower pH.
Neil, there was an additional question for that, and are there any biochars that offer some sort of liming agent as well in terms of increasing the pH? I think the question just disappeared. Oh, here,
right.
You get a liming value,
Yeah, right. So many of these biochars, we do get a liming value from them. So they are more basic in nature, many of them. So I don't know that they often calculate it. You know, when we add a fertilizer, we can get its potential acidity or its potential basicity in the fertilizer world, and the potential basicity tells us how many pounds of liming value we get per ton of that fertilizer. But at any rate, we can take into account, at least with preparing our own mixes or working with a supplier to prepare our own mixes, to reduce the amount of liming that we have to add, because we get a liming benefit of many of these biochar materials. Does biochar addition affect basil quality, its foliar composition, its aromatics, in addition to its size? I will say we didn't study that in particular detail. Anecdotally, when we smelled the basil, these bigger plants, we also smelled, we got kind of that expected basil smell, and there was more of that with these bigger plants. So I think that bodes well. Our thought was that they were very commercially viable, and they had at least, you know, like normal levels of aromatics. Right, found the company, not the product, ClearStack, and familiar with Biomass Control as a sister company who pyrolyze wastewater solids. Yes, and I'll see if I can look up information on the specific furnace that we used from ClearStack itself. But they've got a range of wood burning furnaces that are not for biochar preparation, but they also have many different biochar type products. So I know the animal waste industry is one of them. Good, and thanks for the information, Alex, on the German biochar boiler as well. Then on vegetable plants and amended mediums, were there any nutrient analysis or flavor assessment? Right, so as I mentioned, unfortunately, there was not like a scientific way of analyzing the nutrients or the flavor. In terms of the nutrients, like the mineral elements, the biochar amendment led to somewhat greener looking plants. And so the overall fertility, at least nitrogen, was quite good with the biochar incorporation. Then did the commercially purchased biochar have a compost or other amendment that would give it a higher nutritional value before adding additional fertilizers? From what I'm aware of, the commercial one was just a biochar. It wasn't like a biochar that had been incubated with compost or something, or co-kept with compost. But it did seem to, for whatever reason, be slightly higher in the salts and fertility based on like crop sensitivity to it. I know there's other bodies of research where people will look at preparing like biochar with manure-based compost or vermicompost, for example, and that can be a very nice fit. So the biochar can with its nutrient retention capabilities, can pick up some of the water-soluble nutrients from compost and kind of hold on to them, and you get kind of a nutrient-enriched biochar. I referenced a paper when I showed the nutrient analysis of the maple biochar. That particular paper the postdoc that worked on this project, she collaborated with people at the Rochester Institute of Technology, and they, in that 2018 paper, what they looked at is using biochar to filter yogurt whey, which is a waste product. So in upstate New York, there's a lot of production of Greek yogurt these days, and the whey, the unwanted liquid, has nutrients. It's also quite acidic, and it's kind of this waste product. And so incubating that with biochar led to more nutrient-laden biochar that the biochar itself had plant growth promoting properties, kind of like I mentioned, that nutrient-laden biochar from the yogurt whey had even greater growth promoting properties. And I can post a link to that paper in the chat in a little bit, or the Q&A.
Yeah, I think we'll do one more question and then we'll go ahead with Debbie's presentation.
Sure, sounds good, right. And then I can answer to more of these questions in the Q&A by text. Yeah.
Okay.
So did we age or mellow our biochar or charcoal before placing the seeds in the pots? By letting the biochar get rained on several times will help lower the pH and make the raw charcoal less harsh in the initial application. We did kind of age our biochar. I think maybe not so scientific about it. We had it stored outdoors. It was covered typically for 1 to 3 months before we used it. So when I get the rainwater point, that makes sense. I think we were worried about losing nutrients, or we kind of wanted to see what we would get from biochar that hadn't been leached out with rain. But that's a good point.
All right.
Good.
All right. Thank you so much, Neil. I am jealous of the response of your plants. It looks like they responded to the biochar a little different so far than what we're seeing in our trials.
Okay, right. Annual vegetable crops are good ones to grow in these crop growth trials. Yeah. Cool.
All right. And thanks for checking out the rest of those questions. And then we'll do more Q&A at the very end of the webinar as well. All right.
Perfect.
So Debbie, why don't you go ahead and take over the screen? So our next presenter is Debbie, or Deborah Aller. She is the Agricultural Stewardship Specialist and also soil scientist with Cornell Cooperative Extension of Suffolk County. And she's going to discuss some of the trials, the container portion of it that we've done here on Long Island with biochar. Thanks, Debbie.
Great. Thank you, Mina. Can you hear and see my screen all right?
Yes. Yep.
Okay. Awesome. Well, hi, everyone. I think many of you have heard me talk already in this series, but today I'm going to be just talking a little bit about the trials that Mina and I have been doing on Long Island, New York, so a slightly different part of New York than Neil is working in. And my title is almost exactly the same as Neil's, but I'm going to be talking about obviously a different trial and different plants as well. So not vegetable production here, but ornamental crops within containers. So this is based on a trial funded by the Northeast SARE, and it's a 3-year trial. And I'm going to be talking about the container portion of this trial before Mina talks about some of the field aspects of it. So as Mina said, I'm Debbie Aller. I'm also with Cornell Cooperative Extension in Suffolk County. So, okay, not wants to move. Okay, so just to orient you where Long Island is in the state of New York, obviously we're in the US, New York's here, and we are down here on Long Island, very close to New York City. It's a fairly suburban area. However, the eastern end of Long Island in this area where the trial is located is a fairly rural part of the county. We have about 34,000 acres of farmland in Suffolk County and over 500 agricultural producers. Suffolk County itself is #1 in all of New York State in terms of horticultural products sold. So it's a very high value agricultural production on the East End. And we're about 60 miles east of New York City. So we have a lot of high value markets, including the Hamptons and places in the city as well. So this is just an aerial view of our study site in the 5 different locations. This is both done at 4 cooperating farms, and one site here, site 5, is the Cornell University Long Island Horticulture Research and Extension Center. So site 1, which I've indicated in red, and site 5, those are our 2 container production sites for this trial. And then the 3 sites located in blue are our field production sites, which Mina will be talking about shortly. These are just a couple pictures of container production on Long Island. Hundreds of different crops grown. Like I said, very high value. But there are many challenges for our growers in this area. Issues with nutrient leaching, which might be leading to chlorosis as a obvious yellowing of the leaves on many crops. Various issues with moisture retention, and maintaining porosity in some of these soilless medias. Obviously, growers use a wide range of soilless medias, but they're still having many issues here. And 2 high-value crops, or not high value, this is a high-value crop, boxwood. It's widely grown on Long Island. There are a number of issues with it, including chlorosis. However, it's very popular on Long Island, becoming more popular, particularly because it's relatively deer resistant. And we have lots of deer pressure here. So these are just a couple pictures of the Boxwood Green Mountain cultivar we're using in our trial. The second plant we are using in this trial is switchgrass, or Panicum brigatum, and the cultivar is the heavy metal variety. Just a picture of what some of the containers look like for this crop. So we thought some of these plants, they're having issues. We're seeing chlorosis. We're seeing issues with within the media, nutrients leaching out. Growers are complaining about, or commenting, oh, it's not retaining water, irrigation issues. So Mina and I kind of put our heads together and was like, what about biochar? People are asking more about it. Is this something we could use and trial for our growers here? And when you think about all these potential benefits of biochar, which we've discussed in several talks already, and Neil gave a really nice refresher before, biochar might be an excellent way to improve nutrient retention, water retention, help with the bulk density and the porosity of the media itself. So we decided, let's see what we can do with biochar and a couple of these ornamental crops, switchgrass and boxwood. So before we got started, when we've had many questions here of how did I choose the biochar for a selected purpose? So since Mina and I were really interested in the retention properties of biochar, we asked a biochar supplier, could we have a small amount of biochar? And we just did a simple column retention test. So all we were interested in here is looking at how much water was retained by a given biochar. And we also just did a basic measurement of the pH, because as Neil mentioned, and many of you have asked, the pH has a significant impact on a soilless media or a soil in the field. So we simply combined, put about 150 grams total of biochar and soil. So here our control is actually a field soil, a local field soil. This isn't for the soilless media, but since this study was both has a container and a field component, we wanted to have one biochar for both aspects of that. So we were able to get a corn stover biochar, a wood chip biochar, a hardwood biochar, biochar produced from oats, oat husks, and then a very finely ground corn stover biochar as well. And then we just had our control, and we were simply doing a gravimetric weight between adding a known amount of water and then weighing these columns and figuring out what's the difference, which ones retain the most water. And what we found here is that we ended up choosing the wood chip biochar, this hardwood biochar. And that was because not necessarily the water retention was the greatest, because you could see it's less than what it was for oats, but the pH was more neutral compared to the other ones, which were slightly higher. And also one of the biggest things in selecting a feedstock is thinking about locally available sources. In New York, and particularly on Long Island, we don't grow many oats other than for cover crop purposes. So if we're thinking about the practical side of using a biochar, wood chips or a hardwood based product or a mixed wood based biochar is going to be more available for our local growers than corn stover and oats and some of the other biochars on the market. So that's the reason why we chose this hardwood or wood chip biochar. Just a couple properties about it. It's a slow pyrolysis biochar produced at 100 degrees centigrade. It's about 73% carbon. It's a very low ash content and no toxicant concern. So we did do a full toxicant assessment of the biochar in this trial, which is not something everyone's able to do because they cost upwards of $1,500, which is something that Jillian described in her presentation last week. Here you can see rough particle size of the biochar we received. And as Neil talked about or mentioned, this idea of co-composting biochar with a compost or a vermicompost or a compost tea, we decided, can we get some of these synergistic benefits of a compost tea and a biochar? So we decided we're going to inoculate this biochar. So we really wanted to charge the sites of the biochar before we were applying it for a multi-year trial. Would this help with seeing benefits right away, helping with this aging or weathering of biochar in the field? So we inoculated the biochar after weighing it out. So these are some pictures from the field trial. Sorry to steal that from you, Mina. But we did this in general in bulk for what we used in the container trial as well. So we basically inoculated these for 5 days. I've heard people doing it for 7 days, for 2 weeks. So there's a range of different times that you can do. You can inoculate biochar for, and you can also buy it pre-inoculated or pre-blended with other products. So just some pictures of how we inoculated the biochar for this study. And now some treatments for the containers. So similar to Neil's studies in basil and tomatoes, that we used 3 different application rates. We did slightly higher application rates than he had used. We looked at 10, 20, and 30% by volume. And we were using a standard peat and bark based soilless media. And we decided to look at 2 different methods of incorporation because we were not just interested in its ability to, potentially change the media itself, the structure of the media, but its ability to capture nutrients and water. So we looked at mixing the biochar both within the entire media, as well as just using it at the bottom of the media of the container to see, could it capture some of the fertilizer running out and prevent it from running out. So we had 5 replicates per treatment, plus a control for 7 total treatments. We had the same fertility and pest management, both at the research lab as well as at the grower's site, and completely randomized. And this is a 3-year trial from 2019 to 2021. So we're really in the middle of collecting our data. I'll present some initial findings, but realize these are still very preliminary results, and we will be sharing them fully at a later date at the conclusion of the study. So obviously, like I said, we have been studying boxwood and switchgrass. We plan to add hydrangeas as another crop in 2021 for this study. And also, I forgot to mention just briefly, the fertilizer used, it was a control release fertilizer. We used micronutrients and we added a dolomitic lime. Just a couple pictures of a setup of our trial, kind of the measurement. We use a simple volume basis for 1.5 gallon pots, figuring out what percent would be 10, 20, and 30. Mix that in for a mixed biochar treatments, and then we planted 1-year plugs. This is just looking at the panicum plantings there. These are just a couple pictures of what the bottom treatment looked like versus the mixed treatment with our biochar and bark-based media. So on the right, the mixed, you can see it's thoroughly incorporated, and we just mix this up separately in a pot before putting them in our containers. And for the bottom, we poured the known volume of biochar to the bottom, and we pressed that down, trying to avoid any loss of the biochar out any of the holes at the bottom of the pot. So you can see it was kind of just a line of biochar at the bottom. One thing to note that we had issues with applying biochar at the bottom is, as Neil mentioned, wind and weight of pots. It made our plants very top heavy. So we had to find a way to secure our pots because they were very light on the bottom. So something to consider if you're thinking about putting a very light material at the bottom of a growing plant. But we were able to secure these for the trial. Just what data we were looking at, we analyzed the media itself, foliar analysis during the season, looked at the root and shoot biomass. So here's a picture of a root ball, and we looked at the above ground biomass weight. We did periodic plant health evaluations, and this was looking for signs of sclerosis, for pests, for disease, overall plant health. And we analyzed the leachate for total nitrogen and ammonium and nitrate in particular. And we used the North Carolina, or the NC State pour through procedure. As you can see, Ag Stew technician here doing a pour through, conducting a pour through on the Boxwood plant. And we were doing this for pH and EC. So these were done every 2 weeks to gather information if pH and EC were changing over throughout the season as potentially fertilizer was being leached out of the pots. Just some very preliminary results from the first season. And this is looking at the switchgrass at the on-farm location. And really what we're seeing so far is a lot of variability, not really seeing a consistent impact of biochar for panigums. And on the left here, we're looking at nitrate leaching, and this is average nitrate lost from the pots over the 2019 season. And we analyzed or collected the leachate at 4 different times during the season. And here's just a legend of for the treatments. Our controls in purple, biochars, the B is for bottom, the M is for mixed, with our different percent biochar applications. So you can see our bottom 20% by volume biochar was higher at some point, and by the end of the season, they were all pretty much the same. So far, we're not seeing any differences. We're seeing quite a lot of variability. And if we look over here on the right for ammonium leaching, average loss here, we can see that the mixed biochar at 30% had low leaching. But then we look at our mixed rate at 20% by the yellow line here, and it's above a control. So we're seeing a lot of variability here. Once again, we're not seeing any statistical differences between some basic properties of EC, pH. We have no differences in biomass, unlike what Neil was seeing with some of his plants. And plant health, all plants were looking very healthy in the first season. This might, have to do just with the age of the plants. And here we're talking about switchgrass again. We did see very small differences in some of the dry weight of the root ball at the end of the season when we destructively sampled. Now, when we look at the boxwood plants, and this was specifically done at the research lab, once again, nitrate, average nitrate leaching on the left, ammonium on the right. Once again, there's a lot of variability. We're not seeing any consistent impacts of biochar in reducing nitrogen leaching from these plants. So we're still tweezing apart some of the data, but so far there's not necessarily a positive benefit, not a negative benefit, pretty neutral. Similarly, no differences in some basic plant and soilless media properties there. Just wanted to show you some pictures of what the media and biochar looks like when we split some of these root balls. So on the bottom here, you can see the control of just our media. These plants, like I said, were destructively sampled after 1 season. So the roots pretty much filled in that container. Then you can see on the top left, this mixed, the biochar media mix, and this is a 30%, these pictures are 30% biochar mixed. And you can see the color of the media is slightly darker with the biochar as expected. But roots kind of look similar. There's no, visual real differences in the size of the root ball here. Really interesting when you look, you can really distinctly see where the biochar is when it's applied only to the bottom of the containers. This very thick black banding at the bottom. And like I said, we did have issues with the plants toppling over when it was very windy. So we really had to find a way to hold down these plants then. But it's just a really cool visual here. So I thought I'd share that with everyone. And so far, I just started looking at the data from this season, still putting a lot of it together, but I was able to analyze some of the basic properties of the media and the plant so far, that once again, we're not seeing any treatment effects between just the standard soilless media and the various biochar application rates and application types. But the data analysis is still ongoing, and we still have quite a lot of data to look at and to collect all of next year, and hopefully adding in the hydrangea crop, as I mentioned. So please keep a lookout for hopefully a publication in the future from this work, as well as other presentations. And so I just wanted to thank, obviously, my colleague Mina, also doing a great job putting this series together for everyone, and all the AgStu technicians, in particular Andrew Della Vita, who's been the main one helping us collect a lot of this field and container data, and our the 4 cooperating growers we have involved in this project, and also Sarah for funding this project. So thank you, and I'm happy to answer questions if there are any, or we can do questions after the field portion.
Debbie, there are a couple of questions that were asked earlier. So if you want to just open up the box and see and check those out, and I'll go ahead and I'll share my screen.
Great. All right. Is there any information on biochar that has had repeated use? The biochar in studies was first time, one-time uses. Does current practice not allow for repeated use of soil mixes? Could biochar mellow mixture over time in soil mix? I'm guessing this question is getting at biochar aging or weathering of biochar, kind of looking at biochar over time. There are a lot more, so I'll do my best to kind of answer this. And I'm not as informed about the horticultural side of actually biochar applications. This is my first study on that end. I've done a lot more with field applications of biochar. But there are more and more studies coming out looking at this aging or weathering effect of biochar. Something I was very interested in graduate school, because the properties of biochar do change over time. So it's become an important variable to look at. And there are different ways to that biochar ages, and obviously environmental conditions impact the rate of weathering and breakdown. So there are people that are looking both at fresh application of biochar, obviously co-composted applications of biochar, and biochars that have been either artificially weathered through various aging processes in the laboratory, as well as other natural weathering processes of biochar. So there are studies available in various environments, but I'm not sure, maybe Mina or Neil can comment on aging within soilless media or horticultural mixes there. What is the best, I lost all the questions, sorry. Could you please give us more details about methods of incorporation, why you're putting a biochar layer in the bottom? Okay, so within our study for the containers, we used two methods of incorporation. And what I mean by that is, one is we mixed the biochar within the soilless media. So it was thoroughly incorporated in it. So you could almost think about substituting like 10% of the peat in your media with biochar, which is also done potentially as a biochar being a more renewable source of energy or feedstock or material for horticultural use than peat is as a non-renewable product. So one was mixing it in. That was one method of incorporation. The second method was putting it at the bottom. And we did that because within our study, we were interested to see if biochar can capture the nutrients leaching out of the media. So we're going to see, would we get greater capture, you know, almost that idea of having biochar as a filter, or potentially the same idea as a permeable reactive barriers on a much smaller scale, but having that biofilter and its ability to hold on to the nutrients that would have been otherwise leached out of the soil. And like I had showed, we're not seeing any differences at the moment.
And it's possible that we're not seeing any differences because, excuse me, the growers that we're working with, they use a controlled release fertilizer, and these fertilizers release very little nitrogen, you know, throughout the season. So one of the growers used an 8 to 9 month release. So it's supposed to not release a lot of nutrients and make all the entire formulation available throughout the growing season. So I would love to test some other types of fertilizers to kind of compare. And someone did ask if we had a control, and we certainly did have a control that did not have any biochar. And then Debbie, there was a question. So somebody asked about.
Yeah, sorry, it like jumped around.
Yeah, I just wanted to address the water retention. This season, we did look more closely at water retention, and we just did some simple tests, some leaching fraction tests, and then also just, you know, water retention, weighing before and after tests. And it's funny how sometimes the results can be all over the place, but Debbie and I have not looked too closely at that data yet, but we will, and we'll write articles about it and present about it too.
There is a question I see, how do you handle the dust? And I just want to comment on this because there have been a number of questions throughout the series on dust related to biochar and particle size related issues. So definitely recommend, if the biochar is quite dry when you buy it, add some water to it. That greatly reduces the amount of dust. Also, if you're applying or working with biochar on a windy day at all, wearing some type of mask or face covering obviously helps cut down on any dust particles. But definitely just watering down the material before you apply it or work with it certainly helps reduce any dust loss. How are the controlled release fertilizers applied on top of the pot or mixed into the soil media, rate, frequency? So it was incorporated when we potted up the plants. It was thoroughly mixed into the media itself. So just the media, if we had the bottom biochar application, and mixed with the biochar and media if it was the mixed application.
All right, Debbie, I think I'm going to go ahead with my presentation. So if you
Go
want
for
to
it.
just start typing in some of the answers for these,
Yep, I'll keep
great.
keep
Yeah, and then
asking.
we can just address some of the others afterwards.
Yes,
Okay, so
sounds good.
oh, thank you, Debbie.
Thank you.
All right, so I'll go ahead and get started with my portion of the presentation. I'm going to be kind of reviewing some of the trials that we did in the field. I'm going to be giving a little bit of background as well, since we don't have too many results to report on yet, because we still have a lot of data to look through. So initially, when Debbie and I were talking about doing this trial, we were discussing, okay, which plants should we choose? And you know, for the most part, the growers, from their questions, that's how we decided on which plants, because the diversity of ornamental plants is vast. You can grow a lot of different plants here on Long Island. For one of the woody plants, we decided to go with California Privet. California Privet is not native to Long Island. It's native to Eastern Asia, I believe. But it's used a lot here on Long Island. That's me right there. My colleague took a picture of me taking a picture of this hedge because I was impressed with how beautiful it was. It was actually in bloom. It wasn't hedged before it was in bloom, or, you know, cutting off all the blooms before they blossomed. And let me just show you, that's the picture I took from someone taking a picture of me. So I was really impressed with this particular hedge, and I wished all the California privet on Long Island looked like this in the landscape, but it doesn't. So the demand is pretty high for this plant because there's a lot of privet problems. These are just a few examples of what I see in the landscape. This bottom one here is just showing that there's a lot of development here all over Long Island, I should say. You know, even if you have an existing landscape, folks will tear down the house and just completely redo the landscape. And when they do that, they definitely destroy the soil structure. So here I have some remnants of destruction caused by compaction. There was an access road to a house that was being remodeled. Folks didn't understand why. The landscapers and arborists didn't quite understand why. The privet just absolutely died. They even tried to do rejuvenation pruning, failed. And that's because privet does not like what you call wet feet. It doesn't like its roots to sit in soppy soil. And that's exactly what happened here. Very, very compacted, just a little bit below the surface soil, or the surface of the soil, and it died from wet roots. Here at the lower right, this is the type of privet hedge that I see a lot of. So privet that has some dead patches, and the screening quality of it is diminished. So you can see through the plants more than I'm sure neighbors want that screening to be. They want it to be nice and dense. And for the most part, the privet here on Long Island is hedged about 3 to 4 times a year. And it's hedged to in inches of its life, quite honestly. So there's a lot of privet problems here. So a lot of people need privet to be replaced, or there's privet planted against privet. There's just a big demand for it. California privet is a 3-year crop here on Long Island. This is just a small trial that I did a few years ago to test different types of fertilizer. I wanted to see how the plants responded to it. Here on Long Island, we're zone 7B, and privet is hardy to zone 6 through 8. So, you know, I'm beginning to question if maybe privet is starting to feel some heat stress here on Long Island, because our July and, you know, even some of our August can be really devastatingly hot here. I'm a New England girl, so I don't like it to be too hot, but you know, heat stress is certainly a factor that I'm considering now going forward. So here we have a year later with privet. They are considered weedy plants, and that's because they tend to grow very, very fast, and usually nothing bothers them. And here we have in the 3rd year, usually with the growers, they'll grow much more, especially from that 2nd to 3rd year. But I only gave it about 100 pounds of nitrogen. I split that application through the growing season. Sometimes that can be tripled or quadrupled from the growers. Some of the existing challenges with California privet that the growers are experiencing are really frustrating. So for a plant that's considered weedy and fast growing, it's very unnerving when you have some of these symptoms that you see on the screen here. So intraveinal chlorosis, stunted growth. I know it can be kind of difficult to see where that stunted growth is, but these plants should not be this size. And then, you know, a lot of the growers are questioning, well, is the interveinal chlorosis because of a nutrient deficiency? You know, when the entire plant looks chlorotic, it can be difficult to tell. We've done everything under the sun to try and figure out what is causing the stunting, what is causing the chlorosis. We've looked at nematodes, we've looked at deficiencies. So that means soil and tissue testing. You know, we've questioned, is it too much fertilizer? Is it not enough fertilizer? I'm questioning if they're experiencing a lot of drought stress, and so there's injury to the root, so it's not allowing the plant to function the way it should. So a lot of questions, and so that's why we thought this would be a great candidate to test with biochar. Some of the existing challenges in general here on Long Island for production growers, the soils here tend to have low soil nutrient and moisture retention, right? Beautiful sandy soils, excellent drainage, so that's wonderful, but again, that can cause challenges with moisture retention because there's also very low soil organic matter. And we have to also worry about groundwater contamination here on Long Island because everything's so free draining and there's a sole source aquifer underneath our feet that allows us to have access to lots of water, but we really need to be good stewards about it. So the benefits that have been tested in many other trials, you know, we feel would benefit some of our field trials or some of our production here on Long Island. So the increased nutrient availability, increased soil moisture retention, and of course, protecting the groundwater with, you know, decreasing leaching of harmful nutrients. Here's just a textural class of one of the fields that we used in our trial. It's for all you soil people out there. It's a riverhead sandy loam, and it tested to be about 55% sand. And you can see as we were digging down when we were installing our lysimeters, this is the upper portion of the soil profile. And then here we are near the bottom. You know, I feel like this picture here shows the saturation of the color too much because once we got to that lower portion, which was about 25 inches down, it was just sand. It was devoid of pretty much any organic matter and it was pure sand. So our initial installation of these lysimeters set at 25 inches and that porous cup was at about 18 inches. I don't think we collected maybe 3 leachate samples from these lysimeters. So we installed this year much more shallow ones so we could actually collect some of that soil water and be able to analyze it for total nitrogen. The grower rates for fertilizer that they use here in Privet for Long Island, depending on the growing season, it can vary anywhere from 50 pounds of nitrogen per acre to 150 pounds of nitrogen per acre. And so the growers will do about 3 or 4 applications per growing season. So total nitrogen used for the season, you know, increases to potentially even 400 pounds of nitrogen for the growing season. And the type of fertilizer that they use for the most part is a water-soluble granular fertilizer. Many of them use fertilizer that's 1688 formulation, and ammonium sulfate is the nitrogen source. Some of them will use a partially controlled release fertilizer, but for the most part, all the growers that participated in this trial use that 1688 water-soluble fertilizer with the ammonium sulfate as the nitrogen source. Real quick, I'll go over Douglas fir. This is another crop that we tested within our trial. I love Christmas trees, and there are a few growers here on Long Island. And this particular grower was interested in it and wanted us to conduct a trial there. And one of the challenges with Douglas fir is nutrient uptake. So the soil conditions here on Long Island are pretty perfect for growing conifers, but some of the uptake of micronutrients in particular, because these soils tend to be very lean on some micronutrients, can be challenging. And so the growers then have to apply a foliar feed, which is more of a Band-Aid, and it's a quite pricey Band-Aid. So we thought, well, why not see the effects of biochar and if it would benefit in terms of that nutrient uptake, especially in the micronutrient. Douglas fir, which is commonly used here in Long Island or commonly grown as a Christmas tree crop, is a US hardiness range 4 to 6. I had to look that up today because I thought, oh my gosh, these poor plants have got to be experiencing heat stress here. But for the most part, it grows pretty well here. But it makes me think that maybe we should start looking at some other species for Christmas tree production here on Long Island. A little bit about the trial. So we tested 3 field rates and we did it on an area basis. So 5, 10, 15 tons per acre, so not a volume basis. And we thought that would just be an easier application method, especially in terms of having growers adopt it if we did find benefits. And for the control, it was just no biochar. The grower was responsible for fertilization and pest management. And then, we were responsible simply for incorporating the biochar into our experimental plots. It was a completely randomized block design, and the trial started last year and it will end in 2021. The way we actually applied it to the field, to our experimental plots, we bagged it up because that was the easiest way to get the biochar to out to the fields themselves. And then we had 5 rows. Each treatment was represented in each of the 5 rows. So that meant we had 5 replicates. And each experimental plot consisted of 10 plants, and there was a buffer on either end. So we were collecting data from 8 plants. And again, that was replicated 5 times. And we tried to make sure that the area of the field that we chose was consistent in soil type, because on Long Island, you could have something like a Riverhead sandy loam crossing with a Haven loam or a Bridgehampton loam. And we didn't want to have those, we didn't want to have that variability in our trial. We incorporated the biochar after planting. It was our best intention to get the biochar in while the growers transplanted, but that just didn't work out. We so badly wanted to do that because it is so much easier to incorporate it into the root zone, but we had to do it after the fact. And we were very careful when we were incorporating it not to actually injure any of the plants. Data collection for our trial has been pretty extensive. We wanted to try and cover all our bases the best we could in order to test if there was an effect, especially if there was a beneficial effect or no effect at all. Because of that interest in nutrient retention, we installed lysimeters in the field in order to test the soil water, and we only tested for nitrogen. And those results have kind of been all over the place so far, but we need to take a closer look at them. We installed some soil moisture sensors and then also soil water potential sensors. So for the soil moisture sensors, those were put in at a 4 and 10 inch depth. And then the soil water potential sensors were put in at the 4-inch depth. And I'm curious to see if we'll actually get some interesting results from the soil water potential sensors, because they're more in the zone of more in the profile of the soil that actually has the biochar in it. We also looked at soil and tissue analysis. The soil samples we've been collecting all along, but the tissue analysis for the field plants, they're just too little, and we don't have enough plants to collect the amount of leaves that we need to get analysis. But next year, we definitely will be able to compare the nutrient levels within the leaves. We did do some soil health tests to compare some of the physical characteristics, biological and chemical, and we'll do the soil health tests again in 2021 to see if there are any differences. We tested for heavy metals, and for the most part, we didn't see anything, but we'll do that again in 2021 to see if there was any decrease or increase. And then for the nutrient analysis, we'll review a little bit of the soil, but so far we're not really seeing any effects. Which is a little disappointing, but we still have to dive into all the data that we have. We did the plant health evaluations, and that's important because visual cues are what the growers primarily look at. And so far the plants all look the same out there. So that's a good thing. None of them are dying. At least there's no trend to them dying. And then very importantly, also the shoot and root dry weights. And I should have been going through the pictures here as I was talking. So there's the soil sensors, and we put them in at, again, the 4 and 10 inch depth, and those have been a bit finicky. So that's why I'm glad we also installed the soil water potential sensors. So here's the soil water potential sensor, and we just stuck that in at the 4-inch depth, and we coated it with mud in order to get good contact with the soil before it was buried down in there. Just a soil core, this is showing when we took our soil samples, we went down to a depth of 6 inches. This is, I think, a little bit more than 6 inches, but just to show you, this is likely from either our 15 or 10 tons per acre treatment. And, it's not, it quickly gets mixed in with the soil. So it doesn't have that same effect as if you just have a thick layer of biochar, right below that surface of the soil. And I'm wondering if that's what you need. But again, these are just questions since we're not really seeing much of a response from the plants, but we still have 1 more year with this trial. This is a screenshot that I took this morning of some of the leachate data. So some of the soil water data. And I need to move this because it's in my way. So, you know, in terms of the ammonium levels, they seem to be pretty consistent, but Debbie and I are kind of shaking our heads at why the nitrate levels seem to be all over the place. So if you look on the left-hand side, these are just different dates that we collected from the lysimeters in the field. And we learned pretty quickly that when the growers irrigated, it just wasn't enough because these soils are so free draining. It didn't saturate the soils enough in order for that porous cup to bring in some of that soil water. So we would make sure that we pressurize those lysimeters before a rain event. And then we would be able to collect samples for the most part after a rain event. Not always, but usually we were able to. So we need to fully understand what's going on with these nitrate levels because they are kind of all over the place. And sometimes it doesn't correlate to when the grower fertilized. So a lot of reading that Debbie and I have to do. The shoot and root dry weights, we collected the biomass of the plant last October. And before I go over that, let me just mention the effect of the biochar on pH. And we really didn't see any effect. The biochar itself, I believe, was upwards of 8 point something in terms of the pH of the biochar that Debbie and I used. And here we have in that soil that was growing the privet, the pH was, you know, kind of a little bit lower than what it should be for California privet. Literature states that privet likes between 6 and 7 soil pH, though some of the growers want their pH to be this low when they're growing privet because they believe they'll get less chlorosis that way. But again, we haven't tested that yet. So here we are in 2020, these same plots, and the pH has decreased even further. And we took the soil samples right around the same time of year in the fall as well. So I think, you know, the reason why the pH is so acidic is because of the fertilizer. Ammonium sulfate, it's a very acidic reaction in the soil. And this does not surprise me at all. I guess I am a little bit surprised that the treatment that's 15 tons per acre didn't have more of an effect to kind of balance out or neutralize that acidic effect. But again, maybe because it was just too coarse within the soil, we didn't get to see that neutralizing effect within the soil itself. Here's some pictures when we collected the biomass of the plant. So when I say shoot, I mean the upper part of the plant, and then you can see some of the roots. In terms of variability, you know, we collected 5 plants per treatment, and not too much variability in terms of the root mass of privet, but there was quite a bit of variability in terms of the shoot growth, which is just frustrating when you're conducting an experiment. We tried to be consistent in which plants we collected. So we collected the second plant in each treatment. We didn't try to only choose the biggest one. So sometimes the second plant in each treatment was a really wimpy looking plant. So that was disappointing, and we knew that it would skew the results a little bit. Here's the Douglas fir. A lot of variability in Douglas fir because they're seed grown. So we certainly saw a lot of variability in the shoot growth and then also the root growth as well. So there's the control on the left and then the one with 15 tons per acre on the right. An example of the Douglas fir roots, they look pretty funky, especially when you try and compare them to the California privet. You know, do these two types of root masses, are they the same weight? I know it's a little hard to tell. They certainly have a little difference in growing style. These plants are transplanted bare root. So I feel like they put on, you know, certainly a bit of growth this year, or rather in the first year that we planted, this is 2019. But again, a lot of variability, and it's just frustrating, but that's what you get sometimes when you're trying to grow plants outdoors. Here's one that we were harvesting because again, it was the second plant within that experimental plot, and this is a behemoth of one. We felt really bad digging this one out, but it certainly is a beauty, but certainly skewed the results as well. So here's a little bit of what we learned. These are the mean root and shoot dry weights for the California privet. So the gray column, the gray part of the column is the root, and then the white part is the shoot weight. And it looks like there should be an effect on the 15 ton per acre treatment, but it wasn't statistically significant. And that may have to do because of all the variability in the data. I would like to explore this data again. Cornell has a great service where there are statisticians that will help you analyze your data. So I'm going to take advantage of that, and I'm questioning, how I should kind of transform this data in order to be able to see if there are actually any effects from the biochar. But in general, I think it's just too much variability, and you need a lot of replication in the field in order to see differences. Here's from results from the Douglas fir when comparing the biomass. So again, the gray is the root, and the upper, the white is the shoot, the upper portion of the plant. And again, it looks like the 15 ton per acre has an effect, but this data, it was not statistically significant. So bummer, but we still have another year. We are going to collect the shoots, so the upper portion of the plant in 2021. We're not going to try and collect the roots because we'll be digging and digging and digging, and I don't know if there's a fair way to do it. So the data analysis is definitely ongoing. We still have to look at all the environmental data. It takes a bit to summarize that, so please excuse us, we didn't have time to summarize it for today's webinar. I'd like to thank Debbie, of course. Her knowledge in biochar has just been outstanding in terms of helping us, along with this trial. And then, of course, the agricultural stewardship technicians, they're amazing in making sure we get the data on time when we're supposed to. And of course, to the growers as well for allowing us to take over a part of their fields and their hoop houses and being able to conduct these trials. I know that some of you may be looking at the Christmas tree there that is painted. I don't know if your neck of the woods, if that is done, but some of the growers here are painting Christmas trees. And I got a text from one of the Christmas tree growers yesterday, and he said, there's a purple haze on my trees. And I got all nervous because I thought something was going wrong. But then he sent me this picture, and it's because he painted his trees again. So I should have known. But anyway, if there's any questions, we'll take them. Otherwise, we'll continue on.
Yeah, thank you very much, Mina.
Yeah.
Great presentation. There are a few questions that have come in about the soils portion. I'm answering the first one,
Okay.
but if you start at the Privet one, that would work.
Were pre-emergent herbicides used or soil tested for accumulation?
That would be great.
Okay, some of the growers do use pre-emergent herbicides, and yes, in that field where the images were, there were pre-emergent herbicides used. A lot of the growers don't like to use glyphosate when the plants are that young, even though our weed scientists did do some trials here at Long Island, and as long as you're careful and you have the shields, the small plants aren't affected. But even in my own trial that I did when I was exploring the different types of fertilizer use with Privet, I use pre-emergent. And you may be getting at maybe the pre-emergent is what caused the chlorosis, and no. It's not the reason why, at least not that we've been able to tell, because this chlorosis is experienced across all the growers, and they all do different types of weed management. Some of them stay away from herbicides completely, but yet they still have this chlorosis. All right. Is there any correlation of organic matter level in soil? Do you plan to test addition of mixture of biochar and compost to soil at planting or pre-planting? I would love to. In terms of correlation to organic matter level in the soil, in general, the greater the organic matter level, then you have higher micronutrients or just higher nutrients in general, and certainly higher cation exchange capacity. So far, I haven't really found the correlation, I guess, between higher level, at least in the trial that Debbie and I are doing, higher level of biochar rate with increased cation exchange. We're just not seeing that yet, but we have to look at our results much more closely. This year, because we had a bit more money left over, we took soil samples from every single replicate instead of what we had done the year before was just 2 of the replicates in order to stay within our budget. But the way things worked out this year, we were able to take a soil sample from every replicate. So that meant that we have quite a few results to go through. And do you plan to test addition of mixture of biochar and compost? So the Douglas fir Christmas tree farm, those images that I showed you, I had done a pilot study there where I looked at biochar and that was sugarcane bagasse biochar. And then part of it was mixed with compost. It was a 50-50 blend. And so we looked at just straight biochar, 50-50 blend, and then the control. And certainly initial results of that for just the straight biochar, it looked like in the needle retention, there was greater nitrogen levels. but that wasn't long-term. That effect has not been long-term. And that was a very small study because I was worried that the biochar may harm the plants. I didn't want there to be a phytotoxicity because there wasn't much in terms of the literature and biochar effect on conifers, but those trees are doing fine and they're actually for sale this year. So, but I would like to explore that further. How does the 5, 10, 15 ton rates convert to percent of soil? That's a really good question. I'm sure Debbie and I can do the math on that, but I can't answer that off the top of my head. I think going forward, probably the better approach is by volume, where you know how much you're going to add in just that upper layer of the soil. That's the way I did it for that little pilot trial that I did with the Douglas fir initially. So maybe that's the way going forward that it should be done. But I think, more studies need to be done as well. All right, I think I got all. What does it cost to do studies such as these? Oh, that's a good question. I think I'll type in my answer for that one. But, I guess Debbie and I, we got a ton of biochar, 1 ton of biochar to cover all our rates for the 3 different farms and then also for our container production. And Debbie, what did that cost? About $2,000, a little over $2,000.
Yeah, for just getting the bulk biochar, it was about $1,500, $2,000, which included freight and everything. But I think we recommend, obviously trying to source local. If you do have a local biochar supplier or producer. But I think also to just add on to your comment there, Mina, is that we have amazing cooperators on Long Island. And I think whether researcher, contacting farmers in your area or growers, see if they're interested in doing a test and seeing if they're willing to donate some of their time or plants for such a trial. And if you're a farmer, potentially contacting your local extension office or Soil and Water or NRCS if you're in the US, and see if they're willing or you would like to do a trial on your property and see. I think a lot of opportunities exist in cooperating on projects like that. It might reduce some of the costs on both ends for people.
Yeah, and some of the high costs for us are the actual sensor equipment, and you don't necessarily have to do that, but in order for us to answer some of our questions, we needed to have some of those sensors. So one of the other questions, has it been considered that chlorosis could be caused by the deficiency of calcium, causing the lack of mobility of iron/magnesium? Although iron/magnesium levels within the soil could be within correct levels, but the lack of calcium prevents the mobility of these ions. We have looked at many different soil chemical analyses along with the tissue samples, and some growers have perfectly adequate soil calcium and then also perfectly adequate calcium levels in the plants themselves. So that's a good question. I would certainly like to look at that more. I wish I were better at soil chemistry. I wish I were an agronomist, but I'm a horticulturalist. So thanks for bringing that up, and I'll certainly look into that further. The analyses that we've done so far, and they have been extensive, has not shown that calcium has been the problem. At least, it's always been adequate within the plant itself. Okay, so I think we're going to move on now to our final presenter. Bryan, if you're there you are. Why don't you go ahead and share the screen? Hi.
Yes.
Thank you everyone for your questions so far. They've been great. Excellent.
Okay.
All right, so Bryan Sales is an assistant professor with the University of North Carolina at Pembroke. And I found this research that Bryan had conducted, I think just a couple of years ago, right, Bryan?
That's correct.
And so I was super excited when he said that he would be able to present on and summarize his research that he did in blueberry production. So thank you very much, Bryan, for being here with us.
Yeah, I'm glad to be here. Thank you, Mina. As Mina said, my name is Bryan Sales, and I'm an assistant professor of biology here at UNCP at Pembroke. I'm so glad that both Mina and Neil found not really a change in pH with the biochar. So I found something similar as well. And Blueberry is super flattered to be invited to an ornamental talk. So thank you again for that as well. It's a beautiful plant. So Thank you for your interest in biochar and its potential as a soil amendment for blueberry production. Today, I'm going to share with you some of the highlights of what I found while investigating the incorporation of biochar into the soil, both at different rates and methods, and its related effects on blueberry growth and blueberry yield. So I have my email on the very last slide as well if you want to email me after this talk is over. Okay, so the greenhouse studies were conducted on site at USDA ARS, the Horticulture Research Laboratory in Corvallis, Oregon, and the field study was conducted at the North Willamette Research and Extension Center in Aurora, Oregon. Funding was provided by Family Forests of Oregon through the U.S. Forest Service Wood Innovation Grant. Our biochar was provided by John Medema through Biological Carbon in Philomath, Oregon, and the plants were provided by Fall Creek Nursery in Lowell, Oregon. So special thanks to all those people who contributed. Okay, here. Let me see. Having just a little bit of trouble. Oh, there we go. Okay, so blueberry has some very unique characteristics that influence cultivation, fertilizer, and amendment type. So blueberry has these very fine roots that grow in this mat-like structure. You can see here at the top 30 centimeters are 1 foot of the soil. While the roots are very small, they don't actually have root hairs. Instead, blueberry makes these mycorrhizal associations that aid in the nutrient and water uptake of blueberry. Blueberry is often called acid loving because it's adapted to a low pH soil, generally between 4.5 and 5.5. Additionally, blueberry has been found to predominantly uptake the ammonium form of nitrogen over the nitrate form. So in general, blueberry is considered to have this low nutrient requirement compared to other horticultural crops. This likely influenced its sensitivity to soil salinity. And blueberry likes very high amounts of organic matter. However, these growth requirements that are specific to blueberry very much influence the organic matter types that are selected. Okay, so acidic woody materials such as bark, wood chips, sawdust, they're generally used for blueberry production for several reasons. This practice, I think, started originally because these materials were aided in part because they were just very low value materials of unwanted forestry materials. However, they're becoming less available due to some competing markets and they're becoming much more expensive. Additionally, these amendments generally have a low pH and EC. For example, the pH of Douglas fir sawdust, the industry standard for blueberry production in Oregon is about 4.5. Sphagnum peat moss, also a component for substrates and containerized blueberry production, is around 4.5. So the pH is important because iron becomes less available at a higher pH, which can cause this chlorosis in blueberry. Also, as the pH rises, ammonium will convert to nitrate at a much faster rate, resulting in less available ammonium. Let's see, the EC is related to soil salinity. So substrates generally that have lower EC values will contribute less to increased soil salinity. And these materials tend to have a high C/N ratio as well. So that can definitely immobilize nitrogen. As a matter of fact, it's recommended when you use Douglas fir sawdust incorporated into the soil for a new blueberry planting to incorporate an additional 95 pounds of nitrogen in the soil, kind of to offset that C/N ratio. These can also decrease soil moisture, decrease soil nutrient retention, and they will decompose over time, so they require replenishment. And all of these things, increase fertilizer use and replenishment, will result in a higher cost of growth for the grower as well. Okay, so what is biochar? I know that you've probably heard a lot of definitions about it. I'll briefly go through it. It's a product of applying heat to these seed-bearing materials under no or limited oxygen conditions. So any form of organic materials such as wood, manure, agricultural waste, they can all be turned into biochar. The heat application can range widely, generally between 200 and 1100 degrees Celsius. Depending on the type of production. So the biochar used in this study was produced via gasification at temperatures around 700 degrees Celsius. And that gasification process volatilizes nutrients and removes moistures, and it creates this structure that is far more resistant to decomposition than the material from which it was made. So the result is this very porous material with a high surface area and low bulk density. This high surface area can contain large amounts of oxygen containing functional groups that when in high concentration, and I think they alluded to this earlier, because as biochar ages, it can contain more oxygen containing functional groups, which this is what gives that biochar its overall high CEC, which increases nutrient and water retention. So the pH is generally high for biochar, and this one was an 8.5 in the first experiment. So that's pretty alarming for blueberry growers. The EC was about 1.2 decisiemens per meter. Recent work for blueberry indicates that the EC, anything below 2 decisiemens is best for blueberry growth. So in general, biochar looks pretty good except for the pH here. Okay, so we initially conducted a 12-week greenhouse study where we grew legacy blueberry in sandy soil, either left unamended or amended with biochar or biochar combined with the material called Bokashi at both a low rate, 10%, and a higher rate, 20% by volume. So you can think of Bokashi as kind of a fermented type of compost. Why did we choose Bokashi? Well, biochar has been found to work synergistically with other forms of organic matter, specifically nutrient-enriched ones. And in the production of Bokashi, it's pretty acidic, so we had hoped that that might lower the pH of the blueberry, but that wasn't the case, and come to find out it wasn't quite as important as we anticipate. So to determine if any of these soil treatments suppressed root rot, we doubled each soil treatment and then inoculated half of those with the pathogen P. cinnamomi. Okay. Alrighty, so some of the questions that we asked here was, how does biochar impact blueberry growth? At this point, we just wanted to see really, you know, is this going to live or die, because the pH was so high and it's so against the grain of blueberry management practices. Also, does it affect that mycorrhizal colonization? Can it increase it? Is that a benefit for blueberry? And then, of course, does it suppress root rot that is caused by the pathogen Phytophthora cinnamomi? And then how does it affect the soil characteristics in general? So, okay, so here is just an analysis of some of the substrate used. You can see here that the pH, the top row there, for the soil was a 5.7, biochar is an 8.5, and the Bokashi biochar mixture was a little higher at an 8.8. The CEC of the soil was very low, 6.7, so not much nutrient retention. The biochar CEC at this point, it's pretty new, 41, and then the Bokashi increased it up to a 92. CN ratio, very high for the biochar, and about a 57 for the biochar Bokashi. This really I think the nutrients and the nitrogen of that Bokashi really offset the CN ratio here. And then if you look at the bottom here, the soil was particularly a sandy soil, 75%, with lesser amounts of silt and clay here. So, okay. Alrighty, so in this particular graph, this tracks the leaf area in each treatment across the 12 weeks of the study. Plants on the left were grown in disease-free soil, and the ones on the right were grown in soils that were inoculated with P. cinnamomi. Looking at left versus right side of this graph, you can see that the leaf area of the inoculated plants in all treatments were much lower than the plants grown in soils without the pathogen on the left. So this is our first sign that biochar did not suppress root rot. All right, so however, when you look on the left side here, the leaf area of plants grown in soils with 20% biochar, that is the top ones here, the square. And you can see these plants grown in 20% biochar had the largest leaf area. It was not significantly different from the 10% Bokashi biochar or the 20% Bokashi biochar. However, all of those greatly outperformed the unamended soil and the 10% biochar. Throughout the study, we really didn't see much of a reaction from the lower rate for blueberry. Okay. Alrighty, so then if we look at the dry weights here, plant dry weights revealed similar results on the graph on the left. The bars that extend below zero indicate, right here, indicate root growth, and the bars extending above zero represent shoot growth here. Plants grown in biochar had greater root and shoot growth than those grown in other treatments. You can see here the 20% biochar had greater root growth and shoot growth, resulting in an overall greater total plant dry weight, which you can see here. It was followed by the 10% biochar and Bokashi, which was significantly greater than the soil only, and the 10% biochar actually, the 10% biochar here actually reduced it just a little bit, but not different than the actual soil only treatment. But again, we really didn't see an advantage in using the lower incorporation rate here. All right, if you look on the right here, you can see everything that was grown in inoculated soils. They all developed disease symptoms and they all had a much lower plant dry weight. So this is just another indication that we did not see any type of disease suppression mechanism here. As a matter of fact, we started to see symptoms earlier in some of the biochar treatments, and I think it was probably a result of higher moisture retention. So we did not find it an advantage for disease prevention. pH. So this graph shows the pH of the soil solutions, which was monitored weekly through the 12-week study. At week 1, the soil only treatments, right here, it was lower than the rest. Remember, the pH of the soil only was a 5.7. And the other ones all started higher, but the soil solution was lower than the actual amendment. And then as the study went on, you can see the pH and everything dropped. So that by the end, everything fell pretty much within the range of that recommended pH for blueberry. So in this particular situation, we did not find pH to be a problem for blueberry production in using biochar. So. Okay, something that's interesting, we did find that biochar in this particular study increased mycorrhizal root rot colonization. Biochar has been found to increase mycorrhizal colonization, specifically in sandy soils. That might have been why we saw this reaction. Furthermore, things like nutrient availability, is the plant actively growing if you have something in the field, timing, the climate, there's a lot of different things that might affect the mycorrhizal colonization. It can go up or down. So in this situation, we found really high colonization rates. Okay, then we use these root assays to quantify the percent root infection of the pathogen P. cinnamomi. And as you can see here, all of the plants that were inoculated did become infected, while the non-inoculated remained disease-free. So this confirms that biochar did not offer any type of disease mechanism in this study. So. Alrighty, so here's a picture of plants on the left that were grown in 20% biochar, and then picture on the right of plants that were grown in soil that had been inoculated also with the 20% biochar. So this is just another example of the visual symptoms that we saw. One very interesting effect that the biochar had on the soil was that it seemed to promote aggregation. Notice these smooth round oval shapes in the soil here. This is a highlight, an enhanced picture. You can really see them here. So what that is clay that has surrounded the biochar. So if you just, press this across a white sheet of paper, you can see biochar on the inside of each of these. So this is a pretty interesting effect. It has been reported before in other sandy soils as well. So, okay, so here's a visual under the light microscope of colonization of root by mycorrhizal fungi. The image on the left is of roots grown in biochar amended soil. You can see these coils inside the root, intercellular hyphae. This thing is just littered with hyphae. In contrast, if you look on the right here, this is in the unamended soil, and you don't see hardly any examples of mycorrhizal colonization here. So I thought that was pretty interesting. Okay, so in this study, our conclusions, we found that biochar increased plant growth and mycorrhizal colonization of blueberry. It had little effect on pH and EC, and it had no significant effect on Phytophthora root rot. Okay, so the next study, then we take it to the field, right? So we took out some things, changed some things, and then did a field study. So the objective was to determine whether biochar improves growth and early fruit production this time of northern highbush blueberry under field conditions. So what did we ask? We asked, how does biochar affect those soil characteristics? Does it impact plant growth and yield? Does it in the field affect mycorrhizal colonization? And what is the economic cost of using biochar compared to a traditional Douglas fir sawdust? Okay. Alrighty, so in this particular study, we used northern highbush blueberry Duke. It's very, very sensitive to the environment, so we thought that might be a good way to show any type of effect. Let's see here. So in the field, we grew the northern highbush blueberry, and then our soil treatments were either unamended soil, the industry standard use of Douglas fir sawdust, which is about a 20% incorporation rate. And since the biochar Bokashi blend at 10% incorporation seemed to be a benefit, we kept that, and we incorporated that into the row as well. Then just to see, well, you know, is this Bokashi really doing something? We blended biochar at the same rate with Douglas fir sawdust and incorporated it in the row at a 10% incorporation rate. Now, because biochar is expensive, in order to reduce the cost, we then incorporated our 20% biochar just in the planting hole at planting, okay? We also did that with biochar plus sawdust at 20% and biochar plus that Bokashi blend at 20%, but just in the planting hole. So this is me and some wonderful students here, and this was a 2-year study. So at the end of both years, plants from each treatment was destructively harvested. This is me and some great students that are helping me dig up some plants here. You can see I'm holding a big wad of blueberry roots there. Okay, so what did we find? The top left figure here is a plant dry weight from year 1. You can see that 20% biochar in the planting hole. Where's 20% biochar here in the planting hole is right here. So that's this treatment. You can see that 20% biochar and then the biochar paired with Douglas fir sawdust at a lower rate, 10% incorporated in the row, both had the highest plant dry weights at the end of year 1. Here we see the unamended soil and then Douglas fir sawdust, just to kind of give you a comparison. This time the biochar Bokashi wasn't as effective as it was in the greenhouse. It's possible that the concentration was a little too high, or there's something about it that the blueberry didn't like it in year 1. You can see here that these are the root dry weights, the stem, and then the leaf dry weights. So what we did is plants were pruned to match vigor. So 34 floral buds were left on the larger plants and then fewer buds on the smaller plants. So the graph below is plant dry weight from year 2. You can see that the 20% biochar in the planting hole here, it also had a really high plant dry weight. So did the biochar plus sawdust, again, incorporated in the row, but they weren't significantly different than the soil only, the unamended treatment. And you can see that Douglas fir sawdust did better here. The reason it was likely not as significant of a difference in year 2 is because we pruned plants grown in this biochar in the planting hole and Douglas fir sawdust in the row much harder. And we didn't prune them as hard, and we left more floral buds. So in this year down here, they're putting a lot of energy into fruit production, to whereas the unamended treatment here, it gets to put a lot of its energy into total plant dry weight. So that's probably an advantage in year 2. If you look here to the right, this is yield. You can see that biochar in the planting hole here and the biochar plus sawdust in the row at a lower 10% incorporation rate had the highest yield. And then this figure on the top here, oops, This figure on the top here is a correlation. So at the bottom, you can see plant dry weight in the first year is definitely correlated to fruit yield in the second year. And that's important for blueberry growers because usually they don't get a crop in year 2 because they don't want to overstress the plants. But if you can put on enough biomass in year 1, then that will be a definite advantage for them, and they can get a yield in year 2, which can allow them to get some money back on their investment. Now, if you remember the picture here that we found in the sandy soil, you see those aggregation. This is a picture of a silty loam soil for the field, and you can see the biochar in here, and you can still see the roots growing through it, but you do not see the same aggregation effect that we saw. And that is pretty much in line with some other people who've reported similar effects. Okay, so cost analysis. So we wanted to say, is it worth it to see if it's worth it to growers to invest in biochar? First, you can see in the second column that biochar is much more expensive than sawdust. See here, $76 a cubic yard versus $9 a cubic yard. Douglas fir sawdust is actually sold in units, which is about 7 cubic yards per unit. And those 47 units at $90 a unit comes to about $4,230 per hectare. To whereas biochar, to do 20% in the planting hole, it takes about 2.5 tons per hectare, $800 a ton. It comes to about $2,000 a hectare. So if growers are willing to put this in the planting hole instead of incorporate it in the row, there's quite a benefit to using biochar, but you know that it just depends. Okay, so our conclusions from the field study is that biochar increased plant growth during the first and second growing season. That first growing season, the higher growth resulted in a higher fruit yield in the second. We did not find a difference in mycorrhizal colonization in the field, and that could be because I sampled at the end of every year when the plants aren't growing anymore. So, so they may not have needed that association as much. We did find that Douglas fir sawdust reduced soil moisture and retention and immobilized nitrogen in the soil. So that is not a benefit of using Douglas fir sawdust. And that biochar was less expensive than Douglas fir sawdust if it was grown in the planting hole. Okay, so then we did this last experiment. So you've heard some talk today about how biochar is more effective when it's paired with a nutrient-rich organic matter or something that has more plant-available nutrients. And biochar can be used to remove excess nitrogen from the air and biofilters or from polluted water streams. And it does this because of its high CEC and its entrapment through these pores. So we thought, okay, well, if this happens, could this, if we start, you know, recycling nitrogen in this way more, could this be a benefit for blueberry production? So we identified several different amendments: alderwood sawdust, 3 size fractions of Douglas fir materials, sawdust, shavings, and wood chips, and then biochar and compost. And what we did was, well, what I did was I soaked them for 24 hours in agitation in a ammonium sulfate solution. Then I screened them and triple rinsed them to remove, you know, excess nitrogen. And then I amended them and grew them again with Duke. And I did it with either no fertilizer and then other low rates of fertilizer, 25 and 50 parts per million. So the questions we asked was, how does ammonium enrichment affect the chemical properties of the organic materials? How does blueberry grow in soil with ammonium-enriched substrates? Do plants grown in ammonium-rich amendments require less fertilizer? And then how does growing plants in ammonium-enriched amendments affect plant nitrogen uptake and concentration? Okay, so these are the amendments untreated to the left. This is the nitrogen content and then concentration, and then after enrichment. You can see that all of them really absorbed a lot of nitrogen. the most being at the bottom. If you look at that biochar, it had 4 grams per kilogram of nitrogen, and the enrichment took it all the way up to 30. So the same as the compost after enrichment. This represented about a 750% increase of nitrogen. Okay, so what did we find? We found that once the amendments were enriched, fertilizer rate did not have an effect. So the results that I'm showing you are going to be averaged across all treatments of fertilizer. Again, in this graph, everything below 0 represents root growth here, and everything above represents a shoot growth. So if you look at the root growth, you can see that plants grown in enriched amendments had larger root growths. In unenriched amendments here. So larger root growth here, 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 and here, except for the Douglas fir sawdust, which was pretty interesting. This likely is because these plants are growing roots to explore more of the soil volume, to take up more nutrients, and they were able to put less of that growth into shoot growth. Whereas once these plants were grown in enriched soils, you can see much higher shoot growth here than their unenriched counterparts. So the highest in shoot growth was the enriched compost, but surprisingly, it wasn't different than the enriched Douglas fir sawdust, which is pretty interesting. And the Douglas fir sawdust was not significantly different than the biochar itself. So enrichment was a benefit for everything, and enriching biochar, you can see definitely increased the overall growth. So that was pretty interesting. Okay, so total plant nitrogen content. So you can see here that enriching everything. So these are the unenriched here in the white, and the dark are the enriched. It greatly increased nitrogen content in all of the plants. So nitrogen content was calculated by taking the concentration of the nitrogen and multiplying it times the plant dry weight. So that's pretty interesting finds here. Okay, so our conclusion of that study is ammonium enrichment increased nitrogen concentration and lowered the C/N ratio of the organic materials. Absorbed ammonium was plant available. So that's pretty interesting, because you see a lot of studies where people take these organic materials and they dip them in a liquid solution, and then they measure the liquid solution before and after, and that's how they understand that there's uptake. But it's very rare that they ever actually grow a plant with these type of studies. So it was nice to be able to actually do that and see that the nitrogen that was absorbed was also plant available, excuse me. Okay, so these overall conclusions: biochar increased plant growth and yield of blueberry. It did not negatively impact the soil pH. It increased mycorrhizal colonization in a sandy loam, but had no effect on root rot. Incorporating biochar in the planting hole reduced the establishment cost of blueberry. Ammonium enrichment increased nitrogen concentration of the plants and of the substrate, and it lowered the C/N ratio of the organic materials. And ammonium enrichment enriched sawdust and wood chips increased total plant dry weight and nitrogen uptake by 177 and 460% respectively. So relative to using unenriched amendment. So that's pretty interesting. Okay, so I will leave that up for you. And
Very interesting, Brian. It's an elegant study that you did.
is it? It felt like a lot, so I'm glad you liked it.
Yeah, you went through it very succinctly. There is a question here in the Q&A. Can you access the Q&A?
Let me see here.
While you're accessing it, I have a question.
Uh-huh.
I didn't quite see it. What was the percent soil organic matter when you were doing the field study?
Ooh, it was about, I don't think I had it there. I have it. Can I get it to you? I think it was about 1%. I'll have to check.
Okay.
That field study, it was done in an area where a lot of field studies are done as well. So that could have affected the original organic matter. Okay, so is there a labor cost difference between incorporation of Douglas fir sawdust opposed to planting incorporation of biochar at the planting? Yes, I'm sure there is. These growers They have a very succinct system for commercial production. And I have been told from people who are more familiar with it than I am, that to stop and incorporate it in each individual row would increase costs. Now, that's at this moment, but things change a lot, and there are other avenues like smaller farms and things like that where they may want to take advantage of that. So, but yes, it will increase cost.
Now, I have another question. In North Carolina, aren't there a few producers of biochar in your region?
So I found it really, I just finished a containerized study of blueberry, and I found it a huge challenge to find
Really?
a biochar producer here. Yeah, I found it a challenge. So if you know of any who are interested in moving forward with some more studies, that would be great. Fortunately, when you're doing, I think if you're doing a study, it's always good to source locally, which is really wonderful, especially if you're doing a field study. But I think if you're going to do containerized studies, and that can be done in a lot of different locations, then, you know, maybe using something that's not as locally produced could be somewhat sustainable, depending.
Okay. Are you finding that a lot of the blueberry producers in your area are trying to find biochar and they would like to use it, especially after the results of your study? But
so this study was conducted in Oregon, and I found, and I'm sure that there's a lot of interest, specifically with nursery production for the first few years to growing it, not for actually nursery production for blueberry yield, but just getting these plants available to where, you know, all these different farms can purchase it. So I have had the grower of Fall Creek Nursery reach out to me, and I think that they're doing some studies themselves. So they're very interested in it.
okay. All right.
Yeah.
Really interesting.
Yeah.
So I don't see any more questions. I think we'll just wait and see if any more questions come in. So you said you're doing another trial in biochar production in containers. Can you talk a little bit about that? Or
Yeah, so I'm really excited about it because so much production is moving to containerized production, because when you do this, you get to eliminate all of these soil constraints for blueberry, whether it's pathogens in the soil or pH and just so many things. So now you're seeing all these incredible industries and production that's popping up everywhere in these really severe climates where you wouldn't think of growing anything really. And now they're growing them in these substrates, in these big bags and pots with drip irrigation. And they are just really producing a lot of blueberry. I believe that even though blueberry production has really increased, and I think North America has been responsible for about 50 to 60% of that blueberry production. But because of all of this new production going on in South America and these other places, I think we're only contributing, just dropped down to about 30%. I mean, it's still increased because so much has increased, but I don't think we're the main provider anymore. So I think that's super interesting.
All right. Looks like a couple of questions did come through. So do you see those, or do you want me to read them?
I do. So what do I think about sewage sludge biochar? So I think in general, when you create biochar from such a nutrient-rich feedstock, depending on the temperature, but it's going to have a much higher EC. So that's a lot of challenges with us using like compost in blueberry, specifically compost from manures, because the EC can get really high. So it really depends on how high the temperature is, and just check the EC. You know, I think the EC and the pH. Also, I believe when you get a higher nutrients, you're going to have more ash. I think the calcium carbonate ratio associated, or the liming ratio, was 11% on this one. That's low, right? So you would want to check, you know, how much is it going to raise your pH? So. So we'll have to see. So might be good to look into making a biochar from residue from sugar production from sugar cane. I believe it's called bagasse. It's used to be sold as a container mulch. Yeah, I mean, anything that's an agricultural waste, and locally, I mean, this is the same thinking of how we access agricultural waste locally and use them for composting. You can also take that and make biochar out of it. So what you're doing, you're changing some things, but you're really making it far less easy to decompose, and it's going to have a longer life. And this could be a really big advantage for people in containerized production because they have to replenish it. And that replenishment factor, if it's less with biochar, that can be helpful in the long run. And any experience with microbial inoculants into biochar prior to use in soil or container production? Any guesses about utility of doing so? I think that would be really, I think it's wonderful to use biochar as a carrier system for anything that you want to put into the soil. Microbial inoculants, it depends on what you're growing and how much growth response do you expect from that microbial inoculant. We have blueberries that don't have very much of a mycorrhizal content in their roots. However, they grow really big. So you want to make sure how important that is for you and if it's worth the price of doing that inoculant. I can tell you that we're looking at something pretty cool, I think, right now, is inoculating biochar with beneficial nematodes and putting that into the soil to see if that could be a benefit for growers. So we're working on that right now, and it's looking like the beneficial nematodes are just living really well in the biochar. So I think that's pretty cool. So yeah, I think that would be awesome if you were able to do that. Has the mechanisms of the ash and its reductive oxidative properties for soil nutrients been investigated? I'm sure that it has. I didn't do that.
Yeah, I don't know, Debbie, if you've read anything or if you did any of that in your research. You there, Debbie?
Yeah, I'm
Okay.
sorry, Michael. I don't have that answer.
Yeah, it has been done. I don't know off the top of my head. Something I can certainly follow up with Michael on that question specifically. It might be a good question for Jillian as well, since she does a lot more with kind of mechanisms and just characterizing the biochar. So
Okay.
we can follow up with either myself or Mina, Michael, and we'll try and get you an answer for that. But there's definitely been research on that.
Okay, I don't see any other questions coming in.